Welcome to this introductory course of cell biology. This is part 1 video on proteins. It's an introduction to proteins, so today we will be talking about how proteins are made. Before we talk about proteins, we need to know about the building blocks of the protein, the amino acid. An amino acid is a molecule with an alpha carbon atom at its center, an R group, the side chains, plus an amino group at its one end and a carboxyl group at the other end. At pH 7, both the amino and carboxyl groups are ionized. Because of the alpha carbon, an amino acid is asymmetric, which allows for two mirror images or stereoisomers, L and D. Although most proteins consist exclusively of L amino acids, there are naturally occurring D amino acids. Some can be found on bacterial cell walls, and some are neurotransmitters. So, a protein molecule is made from a long, unbranched chain of amino acids. Therefore, proteins are also known as polypeptides. Amino acids are joined together by an amide linkage, which is known as a peptide bond. These four atoms in the gray shadings form a rigid planar structure, and there is no rotation around the C-N bond. And these two bonds allow rotation, so long polypeptides can be very flexible. The backbone of the polypeptide is called as a polypeptide backbone, apparently, and there are 20 different amino acid side chains attached to this polypeptide backbone. Some of them are nonpolar and hydrophobic, others are charged and can be acidic or basic, and some are uncharged and polar. Okay, so these are the 20 amino acid residues classified according to whether they're positively charged, that means it's basic, or negatively charged, which means it's acidic, or uncharged and polar, or nonpolar. I haven't added the 21st amino acid, selenocysteine, into the description at first, because it is not considered as a standard amino acid, for no apparent reason, I believe. I mean, it is essential to our survival. Many enzymes contain selenocysteine. I think it is considered non-standard because it is made by a process called translation recoding, but it is just my opinion. We won't discuss translation recoding today, but I will explain it at the future videos. So, there is a limited number of possible bond angles in a polypeptide chain. Because of steric collisions between atoms within each amino acid, most of the possible bond angles do not occur. As I said before, the gray shadings do not permit rotation, but these two angles do. Rotation can occur about the C-alpha C bond, whose angle of rotation is called psi, and about the N C-alpha bond, whose angle of rotation is called phi. And the conformation of the main chain atoms in a protein is determined by one pair of psi and phi angles for each amino acid. This is a Ramachandran plot, and each dot represents an observed pair of angles in a protein. The three differently shaded clusters of dots reflect three different secondary structures repeatedly found in proteins. In the next video, we will be discussing protein folding.